Hello everyone. Today we're going to be going over gram staining. The assumption here is that you've already seen our video on smear prep and simple staining. I'll be putting links in the description to those videos, but this should be part of that same playlist. As you can see, we have 4 slides up today. We have your, right here, Sarcina aurantiaca, Neisseria sicca, Escherichia coli, DH5 alpha, and Bacillus megaterium. With gram staining, I would like you to follow along in your lab manuals. For majors, which is BIOL 2421, we're looking at page 51. For the Harleys, for nursing, which is BIOL 2420, we're looking at page 187 of LaBeouf and Pierce. While following along, keep in mind exactly how we're doing gram staining, what the purpose of gram staining is, and its actual rationale. You may also want to follow along with the PowerPoint that is on Blackboard, as that goes more in depth into the actual rationale behind gram staining rather than just the pure technique. I have started here with 4 heat-fixed slides. These are smear preps that we're going to be working with today. We're going to be starting with crystal violet as our stain, and we're just going to start by putting a few drops of the stain on each of our slides. Now, crystal violet is our primary stain. Now, while we wait for this, we take about 30 seconds. Do note that I will be switching up the boxes in between this. At some point, you'll notice the box has shifted from a bluish color to a clear one. That's because Graham's iodine does not play nice with basically everything. So in order to maintain the safety standards here in the lab, we have a specific bin that only uses Graham's iodine and the alcohol decolorizer, as well as a specific biohazard bottle that we use just for Graham's iodine and our decolorizer. So now what we're going to do is we are going to do a very quick rinse of our primary stain. I have off to the side here DI water, and we're going to start with the Sarcina, and we're just going to wash off the extra stain. We do want to use an eyedropper, because if we don't use an eyedropper, the stain can go all over the place. And we're going to do that for each of our preps. And now we're here. We're going to be adding in Graham's iodine. Graham's iodine. We're waiting for that. Let's talk about the rationale behind gram staining. Gram staining is a way of differentiating three different classes of bacteria, the first being gram positive, second being gram negative, the third being gram variable. On Blackboard, you should already have access to a PDF that is a dichotomous key for your unknown project if you are in majors. If you are not in majors, you're just going to have to trust me. That really, this is our first way of differentiating out bacteria. And the way it works is when the crystal violet and the grams iodine meet with each other, they create a complex in the cell wall of the bacteria. And there are some bacteria that have a rather large peptidoglycan layer, which is a part of the cell wall that in this case is going to retain that crystal violet grams iodine complex. Those are gram-positive organisms because they show up as purple after we add in the counterstain. For the gram-negatives, the peptidoglycan layer is not thick enough to stand up to decolorization, which is an alcohol. It shrivels, and that means that the gram iodine crystal violet complex, which is a large molecule, can easily flow out of the cell. These are called gram-negatives. For gram-variable, that's typically organisms like members of the genus Mycobacterium that are resistant to staining and don't stain at all, or they partially stain, and since they only partially stain, you could have a mix of non-stained bacterial cells, gram-positive cells, and gram-negative cells. In the case of the genus Mycobacterium, this is because of the mycolic acid that is located inside of the cell itself. This means that you have to have a different test for it, and that's acid-fast staining. I would recommend seeing the acid-fast staining video when that comes out, or if you're watching this later, see the acid-fast staining video. So now what we're going to do is we're going to take some of our DI water, and we are going to rinse off the grams iodine, just as we did before. Now this is just a preliminary wash to get any of the excessive grams iodine off the slide. This is not to do a test. Now we're going to add decolorizer. And decolorizer, we really have to work quickly. This is the most vital part of gram staining, and really it's the part of gram staining that things can go very wrong very poorly. Since this is an alcohol, it does break down the cell wall, and if the alcohol is left on the sample, even of a gram-positive bacteria for too long, it can burst the cell wall, making the gram stain moot. Here's our decolorizer. And unlike before where I did these in sequence, I'm actually going to do each one individually so that we don't decolorize the stain too much. Typically, you're going to just drop by drop and let it flow. The second thing that I'm hitting it with is just DI water to remove any of the excess decolorizer. And we're going to try it again, just to make sure. As you can see, there are some of these cells that did get decolorized. We'll go over why that might be when we take a look at these under the scope. Next one. Now notice how there's significantly more purple coming off of this one than the other one. That should tell you something about the bacteria. Again, the one I'm flooding with is water to remove off any of the excess decolorizer. And back for a few more drops of decolorizer, followed by a wash of DI water. Now, that was the most time-sensitive of the stains. Now we're going to counterstain. We use safranin. This means that a gram-positive organism is going to appear as purple, whereas the non-grams positive organism is going to appear as a pinkish red. Now, as you can see, we are back to the blue bin. Once again, this is because grams iodine doesn't play well with safranin either. It kind of doesn't play well with anything, and I don't want to have any odd compounds created inside that bin. So what we're going to do is we're going to add some of this safranin. We're going to flood the slide, or at least the area where we're staining. And we let this sit for 1 minute. Now, it does seem like it's only a few seconds for you guys, but I assure you, for us, it's been about a minute. And we're going to rinse off our bacteria. And just with the others, we are going to be using some bibulous paper to blot our slides dry. And from here, we can go take a look at them under the scope. And we're back. I just want to show you one last thing with this. If we take a look, this is another way of setting it up, where you have a positive control, a negative control, and your unknown. This is what the majors would be doing in the lab. And we're going to go through how to set up a gram stain in this way. So just as before, we're going to start off with our crystal violet. And we let it sit. So now that we are done with the crystal violet, we are going to rinse with DI water, making sure to tip it this way so that we can still see our label once we're done. Because I decided that it would be a good idea to stain with crystal violet and use a blue Sharpie. Now that we have that, we're going to switch over to grams iodine. Same as before. We're going to add grams iodine. Boop. Boop. There. And just as before, we let it sit for 1 minute. Now we have a DI rinse. And it's at this point that we have our grams iodine crystal violet complex. And we're going to start to decolorize. Drop by drop. Now, you notice that our markings are now being erased. The reason for that is we use a Sharpie, and the decolorizer is mostly alcohol. So once we're done, we're going to have to relabel what each of these are. Easiest way is just to remember what they are. With on the left-hand side, as you see it, is our gram positive. On the right-hand side is our gram negative control, and in the center is the unknown. And we are going to flood with DI water to do a rinse. And now we're going to go back to the other bin for safranin. And now we are back, and we are going to counterstain with safranin. And let this sit for a minute. And now we're going to rinse with DI water. Now what we're going to do, we're going to put this on bibulous paper and look at it under the scope. And the final thing I want to show is that this is what a slide would look like after you have it relabeled, where you have your positive control, your unknown, and your negative control. And we should be back, this time looking once again at the Leica EZ system. Let's take a look at this. This is our first one. This is Sarcina aurantiaca. And this is one of our controls. This is a positive control, or at least it should be. And we can already tell it is. But we're still going to look in and see if we have some nice stains. This is now the 10 times. We're going to shift up to 40. So here we are at 40 times. Let's adjust our focus. And now we're going to go up to oil immersion. And there we go. Notice how it's a purple grape-like cluster. It looks almost like actual grapes. This is a gram-positive bacteria, Sarcina aurantiaca. Now here we are. We're going to be going through another one. This is a different bacteria. The previous one, that one was Sarcina aurantiaca. That is a coccus. And this is Bacillus megaterium. It's a bacillus. And we're going to bring that into focus. We should be seeing coming up on big old rods. Now we're going to go to 10 times. 40 times. Now, notice with Bacillus megaterium, you can actually see what they look like at 40 times fairly well, which is unusual for most of the bacteria that we have. Most of them do require oil immersion. Now we're going to 100 times. Add the drop of oil. And lock it into place. There we go. Some nice big old purple rods. Let's explore a bit. There we go. We are back here looking at the E. coli. This is the E. coli gram stain on 4 times power. Now we're going to increase to 10 times power. 40 times power. And finally, 100 times oil immersion. Now you notice there are some purple rods here. That is because as part of the decolorization process, the decolorization part is the most crucial part of this stain. If you do like I did here, which is be a little underhanded and not add in enough decolorizer, you're not going to get a fully decolorized group of cells. On the other side, you could go and add too much. Now, we do like to use the drop-by-drop method, as was shown in the previous video. That method is every time you put a drop in, if there's still some color, some purple left, you add another drop until your drop is no longer colored. The issue is that you can quickly go through decolorizer. The other option would be to let the decolorizer sit. If you let the decolorizer sit for more than, say, 5 or 6 seconds, you can really risk over decolorizing. I know in the majors book it does say to let the decolorizer sit for 30 seconds. I do not encourage that at all. You never want to do that because you are going to completely decolorize the cells. Since we are working with an alcohol here, that can desiccate the greatest cells on its own. It's what makes hand sanitizer a sanitizer. Give it enough time, it will do that. So I would not at all recommend using the 30 seconds because you will very much risk decolorizing your cells. And finally, we're going to take a look at the Neisseria sicca, which is another one. As you can see, this one is also, just by the color, you can tell that it's gram negative, though we can look and see that there will be some other interesting things here to look at. Notice how that in this case, it's not as clean and easy to identify as the E. coli was. Now we're going to go up from 4 to 10 times. Let it settle. And come back in. This is 40 times. And now we're going to zoom in once again. Here's 100 times under oil immersion. And let it settle. Now you can see that on this, it's still gram negative because it's still a reddish color, but it does have a deeper red, almost like a reddish purple. And in this case, that's because, once again, I left the decolorizer on a little light, mostly so that we didn't have any issues with overcolorization. And you can see right here are grape-like clusters of Neisseria sicca.